Hello and welcome to another video lecture from MrWatkins.com. Now this video will be on basic monohybrid and dihybrid crosses. Very simple stuff, complete dominance, and that whole genre of working those type of problems. If you have never done any genetics before, you might want to watch the basic genetic crosses video prior to watching this particular video. I'm making certain assumptions that you have some vocabulary that you already know. So let's go ahead and get started here. I'm going to pick a very simple monohybrid cross. Really, I'm just looking at one gene, and so this particular monohybrid cross that we're going to look at what's called hitchhiker's thumb. And hitchhiker's thumb is when you're making a fist here and like you're hitchhiking, the thumb curves back. Mine does not. I'm homozygous recessive for that. Hitchhiker's thumb, the actual gene is dominant, and it's complete dominance. So if we had an individual that had hitchhiker's thumb, so let's say that we have someone who has hitchhiker's thumb, and they're heterozygous for it. And let's say that they mate with someone who does not have hitchhiker's thumb. And I'm going to start abbreviating hitchhiker's just hitch because it makes it easier for me to write. So no hitchhiker's thumb. And if they don't have a hitchhiker's thumb, quite frankly, they're going to be homozygous recessive, but I'm going to go ahead and put it in just so you see. And this would be written in the story problem. So step one in always solving these genetic problems is to come up with the parent's genotype. Hitchhiker's thumb, again, is a completely dominant trait, but we'll find out they're heterozygous, hetero meaning different, so their genotype is going to be big H and little h. I chose the letter H to represent it, to represent the alleles. Typically that's what's chosen, but there's really, for this particular trait, there is no set rule. Other traits there is, and you are expected to use the standard notation for these other genes. Now with the no hitchhiker's thumb, I've already told you it's homozygous recessive. There you go. So big H, little h for this individual. What would be the possible genotypes and phenotypes for this cross? And we want that in percentage, and we're always going to want that in percentage, at least here. Now sometimes they may ask you for like 1 out of 4 or that kind of thing, but you should be able to still figure that out. Percentage is the bottom line, usually what they want, especially in AP and IB. Alright, so step 1 was the parent's genotype. We've got that. Step 2, what gametes can these organisms make? Now we're looking at all the possible gametes. It's not that they only make 2 gametes and they're done. It's all the possible gametes. This one here, it's got 2 different alleles, so it's going to have one gamete that just has a big H, and then it'll have another gamete that's a little h. This organism here has 2 little h's. The only kind of gamete it can make is a little h. Step 3 is to run the cross. Now, a pre-AP student and an AP student and an IB student should be able to have looked at this prior and just said it's going to be 50-50, and they should be able to run the cross in their head. I'm walking through this step by step just in case you need a little practice or you need to kind of see what's going on here. So what we do for that Punnett Square then is I take the one that's on the left, that makes the rows, and that's pretty much the standard convention, but there's no real rule. And I'm going to make a box that allows me just basically to line up the gametes. So I'm just plugging in the gametes here as my rows. And the organism on the right, I typically plug in as my columns, and there's only one here. Now I do know there are some teachers that would make you put 2 columns here with a little h and a little h, and I'm sitting here thinking, why would you want to have a kid go to all that work? It doesn't need it. You're doing twice as much work. We only know it's going to make a little h, and your percentages are still going to come out correct. We fill in the cross here, so we've got a big H and a little h. Each box basically takes one letter, the letter from the row and the letter from the column, and you always put the dominant allele first. Doesn't matter if it's here, if it's dominance here or dominance here, it always goes first. This is a standard convention, and you will lose points. You will not get things right if you don't do it correctly. So I had asked at the beginning what the genotype and phenotypes that are possible for this cross. And here we can see it's going to be 50-50 for our genotype and for our phenotype. We pulled the alleles into the box, and now it's time to do the rest of it. This is going to be the genotype right here. So my genotype is big H, little h, or another possibility is little h, little h. The phenotype, which is the physical characteristic, that's going to be hitchhiker's thumb, and like I said, I'm just going to abbreviate hitch there, and then this one will be no hitchhiker's thumb. We had two possibilities. This is 1 out of 2, or 50%. So when you ask the questions, when the questions are being asked, when you solve what they're wanting or answering what they're wanting, make sure you look and see exactly what they're asking for. In this case, I asked for genotype and phenotype percentages, and it just so happens that my genotype and phenotype percentages are 50%, and there I'm done. There are other videos that go into more detail on a lot harder crosses. That's all for this one.